Okay, welcome to our snow day, sort of snow day lesson or whatever you want to call it. I'm not at school, so I don't have access to all of my stuff, so I'm going to tape right now, and then we'll watch this together live, and then I'll be available for any questions that you have. We were talking about, let me just make sure I'm on camera. I am. Okay, we were talking about genetic phenomena. That's already in your notes, but I'm just reviewing because we've been off. We haven't seen each other for 6 days. Oh, wait, we haven't seen each other for a while. And don't worry about these two pieces of tape. That just reminds me of where the camera is so I know what you guys can and cannot see. We were talking about genetic phenomena, and this is all the stuff that Mendel didn't know, but then affects the patterns of heredity, or how information is not only passed from parents to offspring, but how it's expressed in the offspring. And Mendel didn't know a lot of these things, and we did a few of them. I can't remember how I was labeling, so if this is incorrect, just bear with me. But the first one was incomplete dominance, just reviewing. And you already have this in your notes. And we said the key word for that was blend. So instead of having two specific outcomes, now you have the option for three outcomes. So if you remember in our Snapdragons, the outcomes were dominant was red, recessive was white, and because it was incomplete, there was a third outcome. They actually blended and made pink, okay? So that adds, if you recall, these genetic patterns, all of them are going to add variety to the genetic code, and more importantly, the phenotype or the physical expression of that code. Now, the next one we were working on was codominance. Codominance. And codominance, we said, here was the key, is that now you have two or more dominant alleles. Just reviewing stuff that's already in your notes here, okay? Two or more dominant alleles. I'm going to slide this up on the screen a little bit. There we go. And we said that those dominant alleles are equally dominant, right? Dom is for dominant. They were equally dominant. So if two dominant alleles appear together, we said that they would be expressed equally. Whoops. They would be expressed equally. In our example, just reviewing, our example was the roan coat, and I was doing the red roan in horses. The roan coat in horses. Now, we said that there was a capital R, that's capital is dominant for red. So red hair in these horses is dominant. And then we said there was a capital W, which was white. White is dominant in these horses, and we've never said this before. There are two dominant genes for hair color. So we did a little Punnett square. I'm not going to redo that. I'll just kind of show you. We had a little Punnett square, and then in each one, we had R, big W, and we've never seen that before either. So what was the outcome? There's no blending this time. Remember, blending is incomplete dominance. We're talking about codominance, coming back up. If the offspring was capital R, capital W, you have to go to this region right here. And because they're equally dominant, they're going to be equally expressed. So if you look at the hair on an offspring with capital R, capital W, they're going to have both. No blending. They're going to have red hair and they're going to have white hairs. Some of the hairs are red, some of the hairs are white, and on average, that expression, because it's equal, will be roughly 50-50. Now, I will admit, in the outcome, it's not an equal distribution. Okay, so let me do this. Equal, this is kind of new, so you can add this to your notes. And then I'm going to put a question mark. Is the distribution necessarily equal? Well, the answer to that is no. And what I'm going to do is, when we get to this point in class, I'm going to pause right here, and I'm going to show you some pictures. I'll have those ready to go, and I can share those on the screen for you. So I'm going to pause right there. We'll go over the roan coat of horses, and then I'll tell you a couple more things. So I'm going to hit pause right now, and then we're going to come right back. Okay, and we're back. Now let me come back here. Now we're going to move on to the third one, C. If it's C, I don't know if it's really C. And that's the new one for today. And this one, a genetic phenomenon, is called multiple alleles. And this is just the tip of the iceberg for variety in the genetic code. Not only the genotype, what you physically own as genes, but specifically in the phenotype or the outward expression, okay? Now, multiple alleles, okay, I'm going to write this out. The key thing, multiple alleles, okay? We're going to have 3 or more alleles. Remember, an allele is any different form of a gene for the same trait. I'll say that again. An allele is any different form of a gene for the same trait. So now we're going to have 3 different forms, 3 or more different forms of a gene for the same trait. 3 or more alleles for the same trait. You guys should be taking notes right now and staying off your phones. Okay, now again, all of these things are way easier to understand if you have an example. So we're going to do, and I love this one because we're going to talk about ourselves. Blood type. Blood type. Now, just some highlights here. When we talk about blood type, relative to this concept, multiple alleles, we will be talking about specific proteins. I'm going to put a question mark there because we don't know what they are. And those proteins will be riding on the surface of an RBC, which is a red blood cell. Okay? Now, these proteins are actually markers. And you're going to learn about this next year in 9th grade with Mrs. Walzer when she talks about cell surface proteins. The cell surface proteins on the surface of a red blood cell, are markers, and they provide identification. And this identification becomes very important to your immune system. Because remember, your immune system has the ability to monitor what cells are inside of you, and I can assure you, if your immune system identifies the markers of any cell that doesn't belong inside of you, anybody know what happens? Wait for it. Those cells get killed by your immune system. So all of the cells inside of you have these marker proteins to identify them as part of you. Red blood cells are no exception. Now, through those markers, we create something in humans called blood type. Okay, now let's do this. We're going to come right here. We're going to do blood types. This is something that you all know. Now, obviously, if we were in school, I would question you about this and ask you, but we're not going to be able to do that. Now, there are 4 human blood types. Those blood types are A, B, AB, and O. Those are the 4 human blood types. Now, I'm going to put right here the RH factor. I'm going to put it in parentheses, and then I'm going to leave question marks there. That is a separate protein. We'll come back to that. That is a separate protein marker. So when I talk about blood types, I'm only talking about the markers that identify these. When I talk about blood types plus an RH factor, that would be plus or minus. So some of you are A positive, some of you are A negative, B positive, B negative, et cetera, et cetera, et cetera. Okay? Now, so that's a different, separate thing. We'll talk about that later. Now, let's stay right here in this box. So you have 4 outcomes. There are 4 phenotypes. And those 4 phenotypes are created by 3 alleles. These are the genes, okay? Those genes, they include the alleles, the genes you could get are called A, B, and lowercase I. Those are the 3 genes that create these 4 blood types. Those are the 3 genes that create these 4 blood types. Okay? Now, notice here we have capital, capital, lowercase. Let's start with this lowercase. Since this is a lowercase letter representing a gene, what can you say about a lowercase letter representing a gene? Oh, I know. It's recessive. It's recessive. But wait a second. We got capital A, capital B. What does that mean about those two genes or those two alleles? They are dominant. Both of them are dominant. Oh, look at this. These two alleles are codominant. That's going to be really important. Okay, so you have to know, I'm going to bring this back in, you have to know what codominance is to understand blood typing as it relates to multiple alleles. Now remember, in Mendel's world, there were only two alleles, purple or white, tall or short, green or yellow, round or wrinkled. But wait a second, in real life genetics, sometimes there are three or more. Look at this, 3 alleles. And instead of getting 2 outcomes, look what we get. We get 4 outcomes. Now to really understand blood typing, we're going to go to a little chart here. And I'm going to start this chart, and then you guys are going to finish it, okay? Now on your chart, I'm going to try to keep it in what you guys can see. I'm going to draw a big block, the full length of what's visible here. That should be visible. Okay, and then I'm going to divide that block in 4. So I need 3 lines here. I'm going to go, it doesn't really, the lines don't have to be perfect, but I'm going to try to make them relatively equal. Like that. All right. And then I need 4 rows. So how many lines did I have here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Yeah, there's 15. So I'll just kind of rough it in here. So I need 3 more lines going this way. Oh, that's good enough. I don't know if you guys can see that. That was pretty good. All right, so everybody should be drawing that. I'm going to give you a second to get caught up there. I can always hit pause when I'm playing this video, so don't worry about that. Okay, everybody with? And I want everybody to do this by hand. I don't want you to sit there on your phones, not behind your avatars, doing nothing. I want everybody working with me. Okay? All right, so here's what we're going to do. We're going to do blood type here. Blood type there. That's the phenotype, what you actually have. I'm going to put genotype here. I'm going to put the protein marker. That's what blood type is all about. And then I'm going to put antibodies here. Because your body is already set up to make sure you cannot exchange blood products. I don't know how this happened on our planet, but it's one of the things that blow my mind. And I don't have any putty to fill in the gaps here. So don't even ask me how to explain it. I don't know how. So we got blood type, genotype, protein, and antibodies. Okay? All right, so let's do a couple together, and then I'm going to leave some mystery and see if you guys can kind of figure out the rest on your own. But you have the advantage. You can look this up on YouTube, right? Okay, so the first blood type, we're going to go, let's say that you have blood type A. You have blood type A. What possible genotypes could you have to get A? Now, don't forget, the alleles, your potential alleles are A, capital B, and lowercase I. That's all you have to pick from. So if you want to get A blood type, it's based on the expression. So watch this. If I had 2 capital A's, guess what your blood type would be? A. That's it. A, capital A, those are both dominant. They're both the same. You get blood type A. Now, there is another one. Out of these 3, and I know we can't interact with each other, but out of these 3, thinking ahead, what other possible combination based on previous notes would work? Watch this. A, little I. Now remember, little I is recessive, capital A is dominant, and Mendel's still right here. If you remember, when the dominant and the recessive are together, whoop, the dominant masks the recessive, and you only see the dominant. So if you had big A, little I, you would also have blood type A. Okay? Let's go to the protein. Now, the protein is the marker. Watch this. The protein is the marker. And here's the cool thing. If you have A blood type on all of your red blood cells, guess what protein you have? A. You have A. Yeah, I'm not kidding. You have A. Now watch this. Antibodies. You will have anti-B. Anti-B. Now watch this. I'm going to come down here. Let's say that you have A blood. Okay? And I accidentally put B blood, put B blood inside of you. I give you a pint of B into a person that's A. Guess what your A blood will do to that B blood? Okay, so if you look right here, A blood has anti-B antibodies. So it only wants A. So if I take A and I put in a pint of B, your A blood will do what? It will attack and kill. It's a process called agglutination. That's a U. And what happens is red blood cells start to stick together because of the attack, then they can't carry oxygen. If I give you too much B blood, you suffocate from the inside out. Yeah, not good. You are not allowed to just mix blood types willy-nilly on this planet. In fact, if you got into an emergency situation, emergency medical situation, and you are A blood type, what type do you really want? A. That way your blood won't attack the new blood. Okay, now let's do B. Okay, let's do B. So if I go to genotype, obviously capital B will give me B blood type. Okay? What else would work? Same thing. Dominant B over lowercase recessive little I still gives you B. What protein would you have? You would have B. Everybody see a pattern developing here? And what antibodies would you have? You would have anti-A. Now, I hope you know I'm leaving out a piece of instruction here because I want you to learn how to think. But I will tell you that these two rows are correct. Okay? Now, here's what I want you to do. And make sure, you might want to redo this on another piece of paper. So in case you make mistakes, you can come back and fix this later because we're going to finish this chart in tomorrow's discussion and then talk about our H factor, kind of wrap that up. And, we're kind of getting ready for a test here pretty soon. I'll keep you posted. But I would like you to finish the chart with no other instruction. I just want to see if you can figure it out. I'd really appreciate it if you do it by yourself so I know what you know. But you can work together. I mean, obviously, we're in the Chinese virus online version of school. So, all right, so the other blood types are A, B. I want you to figure out the genotypes, the proteins, and the antibodies. Okay? And then the last one is O. So I want you to figure out the genotype, the proteins, and the antibodies. Some of you are going to see this, some of you won't. It's okay. I want you to do the best you can. All right? Now here's what I'm going to do today. I'm going to post an assignment. And then I want you to finish this chart. I'm going to post the chart. You're going to finish the chart. You're going to take a picture, attach it to the assignment, and submit. Please do not submit if you don't attach your work. It just wastes my time. Okay? All right? And listen, this is the part of the program where I want you to think. It might be a little uncomfortable. Try to put it together. I gave you 2 examples. I will admit they were the 2 easy ones. Gets a little bit more complicated, but follows the same theme. And remember, this is the only alleles you can pick from. So I'm going to end the video right there. Any remaining time, I will be online with you and I can answer questions. Okay?